Okay, so let's get started.
Yeah.
Today we're very pleased to have Peng. Before we get into the seminar, so we are going to give you a quick overview of DASHU and also our upcoming events. So DASHU is an established 501c3 nonprofit organization. We have quite a few strategic partners to do things together. The membership is free. And it's a young organization founded 3 years ago. Everybody volunteers, so none of us get any salary, so such that all fundings go to serve the community. The goal is to provide a virtual community for people interested in data science to promote research and education in that area. We always welcome new members, new volunteers. If you're interested, go to dashu.org. You can contact us and send us an email, or you can contact me after the meeting. We organize international conferences, monthly journal club, as you hear today, technical seminars, and many other things. And for people who are not speaking Chinese, why DaShu? What does that mean? So DaShu in Chinese is a pun. It means big data, big number, as well as big tree. And tree in China symbolize community and community service. We have a saying, we say the earlier generation planted trees, so later, many generations later, they can enjoy the shade and the community around it. So that kind of represents our kind of a principle why we started this organization. So just want to let you announce a couple of events coming up. The first one is April, is an on-site event at Airbnb. So this is most relevant for people who are living in Bay Area, since this will be a very interactive session. So it's on-site only, unlike today's event, that is virtual and on-site together. So that event is on-site only. Is next Thursday in the late afternoon, early evening time at the Airbnb downtown office. So we're going to talk about technical aspects of data science in market dynamic and pricing, as well as the soft aspect, how to be a great data science, what are the other soft skills needed to succeed in that area. And free dinner will be provided, but do register early. I think we only have limited spots, so may run out fast. So if you are already dialed in here, you are automatically a DaShu member. You should have received the announcement. But if you have questions, you can chat with me during this talk or follow up with me towards the end. Also, then journal club. We're trying to do monthly virtual journal club focusing on interesting technical idea like today. So it's still relatively new. We have been successfully doing 2 already, and today is the 3rd one. And we need volunteers and participants if you want to hear about the topic. If you're doing a research you want to present, that's all great ideas. Just contact us. We are very open. We just want to provide a platform so people can exchange ideas and learn new things. And this is the one we actually already have our April journal club, virtual journal club lined up. So it will be towards the end of April, a month from today, and it will be our accurate, robust imputation method as the input for single-cell RNA-seq data that will be presented by Professor Jessica Lee and her PhD student Vivian Lee, both at UCLA. And also, we have successfully done January, February, as well as today, March, 3 virtual journal club, all received very well. If you have missed it, you can see it's on our website. If you just go to our DaShu website, go to virtual journal club, you can see they are all recorded January, February, and today's one, Dr. Peng are very generous and kind enough, also agree as to record this session. So you can, if you have questions, you didn't hear clearly, you can always revisit the video and the recording at a later time, and it's all free, shared with you. So today's topic, today we're very fortunate to have Dr. John Peng to present to us about genome scale signatures of gene interaction from compound screens, predict clinical efficacy of targeted cancer therapy. That's a very hot topic today. Everybody is interested in doing the drug discovery and development more efficient using big data, machine learning, and algorithmic approaches that can really revolutionize the field and make the experiment much, much more efficient. And Dr. Peng has stellar record. I'm not going to go through it. You can see that from his bio. That is all part of the announcement. But I do want to mention one thing I find extremely impressive. He is trained as a computer scientist, had a stellar achievement and GPA and all that from Tsinghua University and Princeton University. But I see so many people changing from life science to become a computer scientist and do hedge fund, to do trading, to find better clicks and push the right ad to you. It is so refreshing to see a computer scientist that are so talented and brilliant as Dr. Peng Jiang, and care about health and life science and helping patients. That is just very inspiring for all of us doing life science and trying to use our skill set to help patients and everyone to have a better health and manage their disease most nicely. So it's very refreshing and very inspiring to see talented people as Dr. Peng Jiang being with us and exploring in this challenging yet very rewarding field. So without further ado, I'll give all things to Peng. I'll stop sharing. I do want to mention to everyone that because there's so many people dialed in, so I muted you all. Just allow Peng to talk. I'll mute myself too. So hold your questions to the end. If there's something you just burning to ask, chat with me and I will ask on your behalf. And after Dr. Peng Jiang's nice presentation, I will unmute you all so that you get a chance to ask questions more interactively.
Okay, thank you so much, Jing, for your fantastic introduction. And I just feel you really flatter me. And also for my transition from computer scientist to biologist, I have a kind of really mixed feeling about this, and we can discuss this if we have free time today. So it's my great pleasure to share the findings from our recent work with the audience here. So Jing, can you hear me? I just want to make sure everything works.
I can hear you nicely, and I'll monitor the chat to let you know if there are other difficulties encountered by other.
Yeah, just a stop.
Perfect.
Yeah, okay, stop in the middle if anything is going wrong. So the paper that I'm talking about today is just published on Cell Systems at the end of this month. It's about a computational approach to study targeted cancer therapy response and resistance. So the hallmark of cancer comprise several biological capabilities acquired during the multiple-step cancer development. Compared to normal tissues, cancer cells could have different oncogenic alterations of genes and pathways, and these alterations supported these malignant hallmarks. Targeted cancer therapies interfere with each of the acquired capability for cancer progression through targeting each of these oncogenic alterations. The development of anti-cancer drugs is an extremely costly process. On average, it takes at least 10 years for a new medicine to complete the journey from initial discovery to the marketplace, with clinical trials alone take about 6 to 7 years on average. The average cost to research and development of each successful drug is estimated to be $2.6 billion. This number incorporates the cost of failures of thousands and sometimes millions of compounds that may be screened and tested early in the R&D process, only a few will ultimately receive approval. So the overall probability of FDA approval after phase 1 clinical trial is only about 12%. So what happened after this long and expensive process? The drug resistance. In the ideal scenario, you hope the anti-cancer drug you develop could kill the tumor cells. But in reality, the cancer cells are pretty smart. They will figure out many different ways to evade or resist the killing from anti-cancer drugs. Here is an example. Vemurafenib is a very famous anti-BRAF targeted therapy. In melanoma, the BRAF gene mutation will promote the malignance of cancer cells. And people developed an inhibitor called Vemurafenib to specifically inhibit the oncogenic mutation of BRAF genes. In this clinical trial, let me see. So in this clinical trial, they got several numbers. Only about 53 patients would respond to Vemurafenib at the very beginning. In other words, half of the patients, even though they harbor the BRAF mutation, they will not respond at the very beginning. And the average duration of response is 6.7 months. In other words, after 7 months, most of the responders initially will not respond anymore. And the overall survival of this responder patient is only about 16 months. So when people read the textbook of some cancer biology, you may see some fantastic examples from anti-cancer drugs such as Gleevec or imatinib or all-trans-retinoic acid developed by Wang Zhenyi from China. The textbook will demonstrate how fantastic these drugs are, and people can survive without any disease after 10 years. However, these types of extreme examples are really extreme cases, but not a norm. To the best of my knowledge, most targeted cancer therapies in other cancer types, except leukemia, will only have a very short efficacy period. So for example, if you look at their progression-free survival, most of the drug will lose their efficacy after 1 year. So biomarker is a very important part of a treatment decision because anti-cancer drugs might be very toxic and very expensive. So if you can use some biomarker to stratify the patient into responders and non-responders in a reasonable accuracy, you can help the non-responders patient to avoid the high cost of treatment and the unnecessary toxicity from the anti-cancer drugs. This graph is some example of some FDA-approved or marketed genomic biomarkers for several different types of anti-cancer drugs. So you can see that some early examples like Oncotype DX and MammaPrint are developed to identify the high risk and the low risk group among ER-positive breast cancer in early stage. And in recent years, people are still developing biomarkers like this. And in other cancer types, such kind of a successful biomarkers is very rare because ER-positive breast cancer normally get a very good outcome. Even though you just predict every patient to be a low-risk patient, the accuracy will be 90%. But in other cancer types, the prediction is much harder. So in summary, that people start to develop some cancer genomic biomarkers, but still there are still a lot of unsolved problems. Besides the treatment decision, cancer biomarkers is also very important in clinical trials. So this is an example from the Checkmate 026 clinical trial on non-small cell lung cancer hosted by the BMS company on anti-PD-1 immunotherapy. So they include lots of lung cancer patients and compared the efficacy between their anti-PD-1 immunotherapy and the conventional chemotherapy. And unfortunately, after many years' effort, they finally realized that the immunotherapy antibody they developed, which is a nivolumab, does not work better than the conventional chemotherapy. So here I have a question for the audience to think about. Does anyone know how much money does BMS lost on this single one figure? So there's only one audience on site. Okay, so this is the data. On Friday, 2016, on that Friday, when BMS announced this negative result, the BMS stock share dropped about 16%, which is $12 per share on the Friday. And if you look at the stock price of BMS, you can figure out there's a really disastrous event when they release the negative result. However, in contrast, the Merck conducted a similar clinical trial, but instead, they only selected 1/4 of patients with an extremely high level of PD-L1. And when they used the biomarker to select patients during the clinical trial, you can see that their anti-PD-1 antibody works much better, or I wouldn't say much better, or at least significantly better than the chemotherapy. And if you look at the stock market, the stock price of Merck is raising up. So this example just demonstrates that biomarker is also a very important tool for the pharmaceutical companies to conduct their clinical trial and improve the successful rate. In the ideal scenario, if we have a large cohort of patients, you can profile their genomics profiles and look at the association between the genomics profiles and the patient clinical outcome. And then there will be lots of statistics and machine learning methods you can use to develop a biomarker to predict the high risk and low risk of patients during the clinical therapy. However, this is only the ideal scenario. For most of anti-cancer drugs, the data is only available on a very small scale. So here is an example. Normally, what you see from a clinical study of some anti-cancer drugs is a genomic, such as gene expression or mutation profiles of patients, less than 50 patients. But remember, there are 20,000 human genes. So if you want to figure out among the 20,000 human genes, which genes are potential robust biomarkers of the therapy response, this is very hard because the gene number or the feature number is so much larger than the number of data samples. So here are just an example of some recent new data on the recent new immunotherapies. So you can see that the feature selection for most of our anti-cancer drugs is ultra high dimensional. However, alternatively, there are many preclinical models developed to study the efficacy of anti-cancer drugs. For example, the cell lines can be isolated from human tumors and cultured in a culture dish as like a simplified simulation of human cancers. And alternatively, there are many studies that use the mouse model as a simulation of human response to different kind of anti-cancer therapies. And the great thing about this type of a preclinical model is that you can easily recruit a large model number and apply automation technology to conduct large-scale screening. So here is one common type of large-scale screening, which is called automated compound screening. So in the compound screening, this is a normal framework. So people or pharmaceutical companies will first collect a large collection of cancer cell lines. And each cell line represents a specific type of cancer isolated from a certain patient. And then there is also a large-scale compound library with different compounds representing the small molecules from different chemical families. And then the modern robotic automation technology can evaluate the killing efficacy of different compounds on each of these different cancer cell lines in an automatic way. And the output of this type of compound screen is a two-dimensional matrix. The different columns are different type of compounds, and different rows are different of cell lines. And for these cancer cell line models, normally their gene expression profiles and mutation profiles are already known. So you can apply a statistical approach to identify which molecular signature is associated with the compound killing efficacy. So these are some examples of such compound screen project. For example, the CCLE collected about 500 cell lines and evaluated the killing efficacy from 24 compounds. This is the public data of CCLE. They have more private data. And there are another two recent large-scale compound screen public cohort. They collect almost 100 cell lines from more than 10 different cancer types and identified the killing efficacy of more than 200 or even 500 compounds. So if you look at the compound screen, this type of experiment is normally done at a very early stage of drug development. But the key problem we want to understand and study is the clinical efficacy of this anti-cancer drugs or this compound in the very late stage, which is after the FDA approval. So the key question that people consistently ask about is whether this type of in vitro screen really have a prediction power on the clinical outcome. In this study, we developed a computational framework called CARE, and the CARE uses the compound screen data as the input and can identify response biomarkers for targeted cancer therapies to predict their clinical response. And CARE can also identify drug combinations for targeted therapies. This is the outline I'm going to follow today. In the first part, I'm going to talk about the key statistic idea behind CARE. So let's look at an example from a compound screen and see what the data is like. In the CCLE compound screen, if you look at the cell lines with BRAF mutations and without BRAF mutations, you can see that the killing efficacy of the BRAF inhibitor is significantly better among the cell lines with a BRAF mutation than other cell lines without a BRAF mutation. However, such association between BRAF mutation and the BRAF inhibitor efficacy is not always true in all conditions. If you further split the cell lines by the status of a third variable, you will observe different associations. For example, when the EGFR expression gets higher, the gap of inhibition efficacy gets smaller between the mutated and other cell lines. In contrast, when the LIF1 gene expression gets higher, the gap of inhibition gets bigger. In these examples, the association between drug target and drug efficacy depends on the status of a third variable. Such relationship is called a statistical interaction, which can be tested by a multiplication term in the linear regression. To better understand the interaction test, we can rewrite the model in this way. By grouping all the variables related with target together, the association between drug target status and the inhibition efficacy depends on the slope, A plus Cp. The interaction coefficient C will determine how the slope changes when the variable P is changing. In the first situation, when C is greater than 0, an increasing level of P will elevate the slope between drug target and inhibition. This case corresponds to those genes whose activation correlates with better drug response. In the second situation, when C is smaller than 0, an increasing level of P will reduce the slope between target and inhibition. This case corresponds to those genes whose activation correlates with worse drug efficacy. Therefore, the coefficient C describes how the slope between drug target and efficacy changes depending on the third variable P. Finally, the computation of chi-score will combine the effect from both the interaction and the base coefficient of variable P together as a student test t-value. This is some example of the multivariate regression from the CARE model. So we put the variables of drug target, another partner genes, and their interaction terms, and computed the CARE score as a t-value. You can see that both of these examples related with EGFR and LIF1 gene expression get a significant t-value. And the CARE score, which is a t-value for EGFR, is significantly negative. And the CARE score for LIF1 is significantly positive. So there are, as I said at the very beginning, there are many large-scale compound screen cohorts, and we can apply our interaction test on each of them and identify for different compounds what genes could be their efficacy biomarkers. So after applying the interaction test on all of these compound cohorts, we will get one CARE score for each human gene for each compound. So here, I demonstrated some examples on the targeted therapies that are commonly profiled across all three cohorts. And there are CARE scores across different genes, but with significant negative CARE scores across several targeted therapies. So you can see that for some of these genes, they are consistently associated with worse targeted therapy efficacy in almost all targeted therapies shown here in most cohorts. For example, AXL. So AXL is a famous example that whose activity will promote drug resistance in multiple targeted therapies. And indeed, our CARE analysis recapitulated this previous knowledge. If you look, so each dot here is one compound studied in each compound screen cohort. If you look at the distribution of CARE scores, you can see that most compounds would have a negative interaction with AXL, indicating AXL as a multiple drug resistance associated genes. For another example, SRC is also a known multi-drug resistance drivers. And you can see that CARE score of SRC is consistently negative in all of this cohort. And interestingly, one previous study identified another kinase as a negative regulator of SRC activity. And this kinase is called CSK. If you look at the CARE score of CSK, we can see that most CARE scores are significantly positive in the exactly reverse direction of SRC, which is consistent with the previous knowledge that CSK is a negative regulator of SRC. After talking about the statistic test procedure of CARE, we will explore whether the CARE score can indeed predict the clinical response of targeted cancer therapies. So as I talked previously, the CARE will model the compound screen data and derive one CARE score for each individual human genes. And for each tumor, when you have their gene expression profiles, we will look at the Pearson correlation between the tumor gene expression and the CARE score as a prediction of drug response. If the tumor gene expression is positively correlated with the CARE score, we would predict it as a responder. If it's negatively correlated, we would predict it as a non-responder. So there are several targeted therapy clinical studies available on NCBI GEO database. We extract those data and evaluate the prediction performance of CARE gene signatures. The first example is the Vemurafenib. We got a BRAF inhibitor, use this procedure to predict the clinical response, and you can see indeed the positive ones, which is the responders predicted by CARE, have a significantly better progression-free survival than the non-responders predicted by CARE. And in statistics, we could use different type of metrics to compare the prediction power from different approach. So I would like to talk that there are many experimental approach that experimental biologists use a lot to study anti-cancer drug resistance. So for example, functional genomic screen is a large class of genetic experimental technologies that you can use to study the drivers of drug resistance. In this type of technology, you use the modern DNA array synthesis technology to synthesize lots of guide RNAs, and each of them can target and inhibit one specific gene. And you can infect the cell pools with this type of DNA oligo through the lentiviral system and assume each cell line has one gene knockdown. Then you treat this pool with either a drug or without a drug, and you can compare the frequency of guide RNAs as a readout. So the readout from functional genomic screens is normally a log-fold change ratio, which is the guide RNA frequency of treated versus untreated samples. You can use this single number on each gene as an indicator that whether this gene will promote or repress the drug resistance. Another type of commonly used technology to study drug resistance is the chronically derived cell line model. So you can take a cell culture. At the very beginning, most of the cell lines will be sensitive to one specific type of anti-cancer drug. And you treat the cell pool with a drug, and after a while, most of the sensitive cells are killed, and the resistant cells grow out. You can compare the gene expression profiles between the sensitive cells and the resistant cells and identify the gene expression biomarkers of drug response. So by comparing the prediction performance of CARE versus the different type of functional genomic screens and chronically derived cell line model, you can see for BRAF inhibitor, the prediction accuracy of CARE is consistently better than all the other approaches. We also evaluate another drug, which is Lapartinib. So Lapartinib is a HER2 inhibitor used in HER2-positive breast cancer. On NCBI GEO, we identified the data from CHRLOB clinical trial. And you can see that among the responders of CHRLOB clinical trial, their correlations with CARE gene signature is significantly higher than the non-responders. So similarly, we use different metrics to compare the prediction accuracy between CARE and other genomic signatures, and CARE significantly outperformed all of others. However, until now, there is a limitation of our analysis because the most clinical trials of targeted therapies do not release the genomic profiles of patients. So this type of data is very limited on some public database such as NCBI GEO. Alternatively, many chemotherapies, many historical chemotherapies has lots of gene expression data paired with the patient response information. And some chemotherapies, like Paclitaxel, inhibit a specific gene, such as the tubulin, the TUBB, as their function and mechanism of action. So in another word, you can treat these chemotherapies still as targeted cancer therapies. and apply care analysis on them. So for Paclitaxel, we identified 3 large-scale patient cohorts from previous clinical studies, and each of them included about hundreds of patients. And you can see that in each of these cohorts, the responders tends to consistently have a higher correlation of care than non-responders. And similarly, the care gene signature outperformed the other signature from genomic experiment. And interestingly, if you look at the prediction power of the gene signatures developed from the clinical studies, the care achieved a very similar performance. The clinical signatures means you use the data in this clinical study to identify some genes associated with response, and use this gene signature to predict the response in another clinical study, which is type of a cross-cohort analysis. So you can see that the care gene signature achieved a performance that is very close to clinical data. We also observed a very interesting result. The care gene signature is computed using the in vitro cell culture screens. However, we used the gene signature of anti-BRAF inhibitor and found that this signature can also predict anti-PD-1 immunotherapy response in melanoma. Both BRAF inhibitor and anti-PD-1 immunotherapy are commonly used therapy in melanoma. You can see that among the anti-PD-1 responders, most of them have a positive correlation with our care signature of BRAF inhibitor. And the prediction metric of care is even higher than PD-L1 or BRCA2, which are some previous known biomarkers of anti-PD-1 response in melanoma. So we further explore the reason for this prediction power from between targeted therapy and immunotherapy. So first of all, we look at the TCGA samples. We apply the care signature of BRAF inhibitor to stratify all TCGA melanoma tumors into predicted responders to BRAF inhibitor and predicted non-responders. So first of all, I want to clarify that the TCGA only tells you the pre-treatment tumor gene expression and the overall survival, but the treatment information of this patient are not well known. So we don't know which patients are treated with BRAF inhibitor at all. But after the prediction and the patient stratification, you observe some very interesting association. Among the responders predicted by care, a higher level of immune cytolytic activity indicate a significantly better overall survival. Immune cytolytic activity is defined by the expression sum of 3 genes. This type of definition is a common indicator used by computational biologists to predict the overall anti-cancer immune response in each individual tumor. So a higher level of immune cytolytic activity normally should associate with better survival. You do see that here. However, among the non-responders predicted by care, you can see that the higher level of immune cytolytic activity does not indicate a better survival anymore, which means that using the TCGA analysis, the non-responders of BRAF inhibitor seems to have a deficiency of anti-cancer immune response. So we apply the gene set enrichment analysis to identify which gene categories is associated with the care signature of BRAF inhibitor. And you can see that, interestingly, top 1 enriched category is called epithelial to mesenchymal transition. It's highly enriched among the negative gene scores of the care anti-BRAF inhibitor signature. So the epithelial to mesenchymal transition is a known biological process that promotes both targeted therapy resistance and tumor immune evasion. And this analysis confirms that why we see the common prediction power between targeted therapies and immunotherapies. So I have to say that there are many previous computational models that people used in the compound screen project. Because if you formulate this problem of identify biomarkers from compound screen data, it's just a very standard feature selection problem. You have an X matrix, a bunch of data points, and lots of gene features. You have a response vector, which is Y. This is the drug response efficacy of different inhibitors. And you can apply some naive method, such as the Pearson correlation or ANOVA analysis to pick the gene features that are significantly associated with the drug response. Alternatively, you can also look at there are a large category of high-dimensional regression approach. Linear regression will fail on this type of application because there are too many human genes, only hundreds of data points. When the number of features is much larger than the number of data points, conventional regression method doesn't work. But the high-dimensional regression method can do feature selection on this type of data. Some famous ones is just like ElasticNet, which is commonly used in previous compound screen analysis, and support vector regression. This is another type of high-dimensional regression approach. So we compare the prediction performance of CARE versus this other high-dimensional or naive statistical method on predicting the clinical outcome of different anti-cancer therapies. And you can see that in general, the CARE signature achieved a better performance than other signatures. So we also do another type of a method comparison based on evaluating the prediction power on predicting the signatures of acquired drug resistance. Previously, all of our evaluation here is testing whether the CARE signature can predict the initial drug response. However, even after the drug response, some resistance tumor clones may still grow out after a while. So we evaluated whether CARE can predict the molecular features of those cell lines acquired drug resistance after the treatment. We collected some studies from a BRAF inhibitor and lapartinib, and these studies established the drug resistant cell lines from chronic treatment of sensitive cell lines and profiled the gene expression profile before and after the drug resistance. The rationale for us to use these studies is because we realized that this type of acquired resistance profile can well capture the previous known biomarkers or regulators of drug resistance. So for example, for most of the famous promoters or famous genes associated with drug resistance or drug sensitivity, you can see the consistent gene expression change before and after the gain of drug resistance in this study. So we use this type of data as a gold standard and evaluate the performance method among different computational approaches. In statistics, we use the ROC curve to compare different binary predictors. So if the ROC curve A is always stay above ROC curve B, we would say the method A is better. So in these two drugs, you can see that the CARE consistently outperformed all other computational approaches. And we also do some variation analysis on putting different kind of tricks in other computational approaches and use the area under the ROC curve as an overall matrix. You can see that CARE consistently outperformed all the other method variations. So until now, I demonstrate how CARE can predict clinical response of targeted cancer therapies. And then I will demonstrate that besides that, CARE can also identify synergistic drug combinations between targeted therapies. So we focused on anti-HER2 therapy lapartinib in breast cancer because the disease focus of my mentor, Shirley Liu and Miles Brown, is on breast cancer. So we selected some top genes from the CARE signature of lapartinib. So we first look at which gene has an extremely high chi-score in the laparnib signature. And we also require that this gene should be significantly upregulated in the cell line's acquired resistance to laparnib. And this gene has to be associated with the pathological complete remission in the Chernobyl clinical trials. There are many genes, like about 40 genes identified from the first 3 criteria, but we only focused on the 2 genes that have known drug inhibitors according to the Pfizer OASIS database. So these 2 genes are AKT3 and PRKT3. So AKT3 was already well known to promote anti-HER2 drug resistance from previous studies. And to the best of our knowledge, we didn't see any report talking about the role of PRKT3 in anti-HER2 drug resistance, so we focused on PRKT3. I first did a simple experiment. If indeed PRKT3 can promote the drug resistance, knocking down PRKT3 should sensitize the cell line to laparnib. Indeed, this is the case. I used a pool of siRNA with 30 different guys to reduce the off-target effect. And after the genetic knockdown of PRKT3, the SKBR3 cell line indeed get more sensitive to the laparnib. The x-axis is different dose dilution from 5 micromolar of laparnib. The y-axis is the fraction of inhibition on SKBR3 cell line. You can see that after PRKT3 knockdown, the curve is really shaped upward towards a better sensitivity. For PRKT3, there are several compound inhibitors developed, and I evaluate each of them. So the black curve is the monotreatment curve of laparnib. And after co-treating with the SKBR3 cell lines with laparnib and this KBMB, which is a PRKD inhibitor, you can see that the treatment effect is significantly better than mono treatment. And the blue curve is the expected efficacy estimated by the Bliss independent model. In another word, if you assume that there is no synergy between the PRKD inhibitor and the laparnib, you would get a killing curve like this, which is significantly lower than the co-treatment curve measured in the experiment. This type of gap means a synergy between PRKD inhibitor and laparnib. So on this heat map demonstrated the Bliss synergy score between different doses of laparnib and PRKD inhibitor. You can see a significant positive synergy. We also evaluated another PRKD inhibitor, and interestingly, the mono treatment of this PRKD inhibitor will already kill the SKBR3 cell line. And however, the co-treatment of them is much more efficient than the estimated active effect from Bliss independent model. These experiments are done on the laparnib sensitive cell lines. How about the drug resistant cell lines? So I derived some drug resistant cell lines from the SKBR3 by chronic treatment. I first treat the SKBR3 cell lines with laparnib. Most of the cells died, but some minor clones survived. And after 2 months, finally, these minor clones grow out. I get a drug resistant cell lines. You can see a significant drug sensitivity gap here. And by treating the laparnib on this drug resistant cell line, you can see that unless you treat the cell line with an extremely high concentration of laparnib, this cell line wouldn't respond at all. But the co-treatment between PRKD inhibitor and laparnib significantly sensitized the resistant cell line to laparnib again. And here is the Bliss synergy score between different doses of PRKD inhibitor and laparnib. You can see that the best synergy between 2 drugs are achieved at a low dose of PRKD inhibitor and a middle dose of laparnib. Besides laparnib, trastuzumab is another commonly used HER2 inhibitor in the breast cancer therapy. So I also evaluated the synergy between PRKD inhibitor and the trastuzumab on the SKDR3 cell model. And you can see that very similarly, that the co-treatment is significantly more efficient than the estimated additive effect. And the synergy score is almost significantly positive between different doses of PRKD inhibitor and the trastuzumab antibody. In summary, I developed a computational framework CARE, which can predict clinical efficacy of targeted therapies using compound screen data. And my result indicated that the data from in vitro screening system could have high relevance on the human clinical response. Remember, it's much easier to conduct compound screen on a large number of cancer models than to conduct clinical trials on a sufficient number of patients. Moreover, my analysis found that the common response signatures exist between targeted therapies and immunotherapies. Last, I also demonstrated that CARE can identify synergistic drug combinations to overcome drug resistance. Based on this result, I can envision several clinical applications of CARE. First of all, since the compound screen data was generated at the very beginning stage of drug development, the CARE framework can identify biomarkers of patient tumors that may respond better to a new drug before the clinical trial. This analysis should help pharmaceutical companies to reduce the failure rate of their clinical trials. Second, CARE could assist the design of combination drug clinical trials. Recent years have seen the clinical trials directly on drug combinations between an FDA-approved drug and another new drug. CARE should be particularly useful for designing such combination trials, which may have a higher success rate than single drug trials. Last of all, I would devote my sincere gratitude to my mentors and collaborators at Dana-Farber Institute and Harvard University. None of my works are possible without the help from my colleagues. Thank you so much for your attention.
So now I'm going to unmute all of you. I understand if some of you are multitasking and clacking your keyboard away. If that's the case, please remember to mute yourself. So I'm going to unmute all so you can ask questions. You should be all unmuted now. Is there any question from the audience? Okay, I will start one, and I think that, oh, is there a question?
Oh yeah, I have a question about the CARE score. So I'm just wondering, like, is this CARE score treatment specific, or is this a CARE score can be like a pretty general for any possible treatment?
Yes, maybe I need to clarify this better in my talk. The CARE score is treatment specific. So in another word, the CARE score will be very different for different drugs. So maybe this figure can answer your question. So these are the CARE scores. And for each drug, it has a unique profile of CARE scores. So in short, it's treatment specific.
Okay, thank you. So basically, in all your examples, you focus on a specific treatment and then talk about the CARE score related to that treatment.
Yeah, exactly.
Okay, thank you.
Any other questions from the audience? Going once, twice, third time? Okay, I'll ask a question that may be relevant to many of you as well. So I know you have a website of CARE. How can I navigate that and doing it for my translational research, if that's my interest?
Yeah, I forgot to mention that there's a, so like for example, like, so this is a website of CARE. You can search a specific like drug name on this website and identify what is a CARE score, which is a biomarker for this specific drug. And the great thing about this website is you can know that what are the functions that are highly enriched on the CARE scores. So for example, you know that from this example, it will say that Laparnib will not probably work so well in the tumors with a high level of epithelial to mesenchymal transition. So in other words, this CARE website can help people to navigate my result on the specific drug you are interested in. And the CARE result can tell you that when this drug will be more effective or be less effective.
Thank you.
Mm-hmm.
Any other questions? Well, I have a second question. You do mention that about some common signatures between targeted therapy resistance and immune therapy resistance. So what are the top genes and their functions in the common signature?
Oh yeah, this is a really great question. So like you really asked the key point that we are working on right now. Let me first find the slides related to this question. Yeah, should be this one. So first of all, I mentioned that the commonly enriched pathway is the epithelial to mesenchymal transition. But there are also lots of other pathways, such as the inhibition of cell apoptosis, inhibition of cell program deaths that are shared between targeted therapy resistance and immunotherapy resistance. So, and we are really following up some of those top hits because this type of enrichment really makes sense, because if the cells have some mechanism to evade the apoptosis, neither a drug or the T cell from the immunotherapy can kill the cancer cell line. So currently, this is our ongoing project, and our lab, the Liu lab, may have some further paper on this topic, hopefully in this year. So please follow up our study.
That's indeed very exciting. We have about 2 minutes left. Any audience have any last minute questions? Now is the chance.
People are so quiet here.
How many on-site people you got? We have
only one.
Well, it's our first try. Yeah. We do have a much larger audience online, and so if there are no further questions, let's do thank speaker, and please follow up other DASHU events. We look forward to seeing you in person or virtually in our community. I will do a quick thank you. Thank you very much for your dedicating your time to support DASHU and give us such an illuminating presentation.
Thank you, and thank you for DASHU for hosting this journal club.
Thank you. So if there's no more question, we'll stop the meeting now. Bye.
Bye.
Thank you.
Thank you.